Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. As we return to a regular programming after a fantastic four series episodes honoring Dr. Marcaron, we would like to take a moment and thank our Dr. GPCR Ecosystem partners for their support. We'd like to thank Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. We have a few announcements to make. We want to let you know that we are revamping the Dr. GPCR Summit, which we ran from 2020 to 2022, and are launching the Dr. GPCR Symposia events. With this series, we aim to bring you, our ecosystem site members, regular and topic-specific virtual free live events. The first symposium of the series will be held on Friday, March 24th, starting at 8 a.m. EST. We will host a full day of talks on Zoom and a 2-hour poster/networking session on Wonder. The topic of this event is recent advances in understanding challenging GPCRs. Our confirmed speakers include Graham Milligan, Ines Liebscher, Ben Myers, Antonella DiPizio, Lucas Gratz, Jake Mahoney, Brendan Wilkins, and Anthony Picard. All trainees are welcome to come and present a poster. There won't be any poster selection. Everyone is welcome. For more information and an updated schedule, you can visit the ecosystem. The easiest way to get to it is to use the links in the footer and look for Dr. GPCR Symposia. You will be able to join us by marking your calendar and becoming a Dr. GPCR Ecosystem free site member. After the event, and if speakers give us permission to record, you will also be able to watch these videos with your premium membership indefinitely. To navigate the ecosystem and find more information about our new events and everything that we do at Dr. GPCR, please use the direct links in the footer to navigate the website. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR, and today I have the pleasure of having with me Dr. Roger Sunahara. Hi, Roger.
Hello, how are you doing?
I am great. Thank you so much for being here. I'm so excited to be able to talk to you.
Me too. I've been following your on Twitter and seen a couple of your casts and they're just fun. They're fun and it's a great opportunity. Thank you for inviting me.
Thank you. Thank you. And thank you for following and enjoying the work that we do. And before we hit record, we were talking about how fun and amazing the GPCR field is. So why don't we start with you introducing yourself, and then we can talk a little bit more about your work.
Sure. I'm at the University of California, San Diego. I'm a professor of pharmacology, and we work on the structure and function of G protein coupled receptors and signaling. I relocated my lab from the University of Michigan. I started my independent career at the University of Michigan. I was there for 14 years, also in the Department of Pharmacology. And it was really there where we sort of got a handle on how to analyze the activity and structure and function of G protein coupled receptors and also G proteins. And it was that move was 14 years ago, and I was really lucky that I was able to bring a lot of people over from Michigan to San Diego to continue their training. Prior to Michigan, I did my postdoctoral fellowship with Al Gilman at UT Southwestern, and which is an amazing experience. And that really is where I got my feet wet in protein biochemistry and enzymology and in structural biology as well. And so I worked on adenylyl cyclase and on G proteins and basically trying to determine the catalytic mechanism of cyclase. And I had a wonderful time as a fellow in the lab. It was certainly an amazing experience not only to be in his lab, but also to be at Southwestern, because it was a very exciting time at Southwestern. And some really amazing discoveries were made at the time. So what got me into Al Gilman's lab was my experience in as a graduate student, and I did my PhD with Philip Seaman, the late Philip Seaman, and that was at the University of Toronto. Actually, I did both my undergraduate and graduate work in pharmacology, both in pharmacology at U of T. But my graduate experience with Phil Seaman was really uh, it was like it was like going on a fishing trip in many ways, and I was just lucky, and we caught a good load. And because of that experience and projects went well, we were at the right time at the right I was at the right time in the right place. And my background, even though it was a pharmacology department, my background in undergraduate was also in molecular biology. So it was helpful at the time to apply more molecular biological approaches to try to tackle some pharmacological questions. And that was my opportunity to really learn about the field, which I eventually built my whole career around. And it was also the time when I got to see all the titans in the field make their seminal contributions. Um, Certainly, watching from a distance what the Lefkowitz Caron labs had done, and what had been done in Europe and what had been done across the country on structure function of GPCRs was really inspiring to see as a graduate student. You just read these papers and you're just mesmerized by them. And I also got to see at the time a lot of the um, former trainees of the Lefkowitz Caron lab also do outstanding work, Brian being one of them, because he's obviously been a close collaborator for many years. But used to, as a graduate student, I followed the work of many of the trainees as, wow, I mean, I can't imagine what it'd be like getting out there and just being on your own and doing all this great work that you learned the fundamentals as a fellow or as a PhD student, and then they got to do their own stuff, and it was amazing stuff. It wasn't just like incremental finding. These are amazing um, uh, contributions, and as a graduate student, to see that was just so inspiring. And so I've been fortunate. I mean, I've been fortunate both in, my research, but also in where I ended up in my training. I think landing in the Gilman lab has certainly affected how I think and how I carry out my research, and just the not only the fundamental knowledge, but Al Gilman was brilliant, and he was a superb protein biochemist. And the collaboration that Al Gilman had with Steve Sprang, who was a crystallographer, uh, opened my eyes up to structural biology and the what you can do with structural biology to help you get at mechanism, and how the biochemistry and enzymology would go hand in hand with structure to really determine mechanisms. And so I think my training in the Gilman lab and also collaboration with Steve Sprang, in particular John Tesmer, who was a postdoc in Steve's lab, and just that experience certainly left an indelible mark on how I think and how I led my independent research career.
Prior to going, well, Brian, prior to getting into pharmacology and in molecular biology as an undergrad, did you always know that you wanted to be a scientist?
Um, it's a funny story. Many people in the GPCR field probably, well, not many, but some people in the GPCR field probably know that science wasn't really my first passion. Um, I grew up, and I still am, an artist, and I was an instrument maker, guitar
Wow.
maker, actually.
Hence the
So
guitar behind you.
yeah, that's one of the guitars. Actually, it's a recent guitar. I made that during COVID. Um,
Wow. Amazing.
but no, I made acoustic instruments,
Hmm.
acoustic guitars. I apprenticed when I was in high school with a guitar maker, and I that's what I was going to do. My mother was an artist. She was a potter. My father was a scientist, professor at University of Toronto. He's a
Hmm.
physiologist. At the same, so I've had this proclivity, at least for the arts, and I really, really, really was I was really in the music scene. I jammed with musicians. I got to hang out with them. I got into trouble with them, and but I got, I loved being in that scene. And um, I also know that it's hard to be an artist. And I watched a lot of my incredibly talented friends struggle just to put a roof over their head and feed themselves, and if they did have kids, feed their kids. And they used to do a lot just to be able to do that to feed their passion.
Yeah.
It was pretty. There were a lot of sacrifices that had to be made.
Yeah.
And I think growing up with a mother who was an artist and a father who's a professor um, probably shaped my long-term goals and thinking, well, it'd be nice to be a professional and then have a hobby as a guitar maker or an instrument maker. Now, what I didn't realize is that actually science is an incredibly creative endeavor. And, I know this now because many of my friends are incredibly creative and are incredibly talented artists in their own way. Some in more formal artistry,
Yeah.
but others in just the way they think, just the way they put ideas together, um, the way their perspective, their stubbornness, their intensity, their focus, their willingness to learn other artists' techniques. I find science to be an incredibly creative outlet for me, my creative outlet. And so even though I didn't get to do a lot of what I thought I wanted to do when I was younger,
Hmm.
I think I was able to express myself and my creativity through science.
I love
And
it. Does anyone own an instrument that you made? Anyone we know? Or what do you do with the instruments that actually you
well,
actually
when I they were I made about 40 guitars when I was younger,
made? Hmm.
and they all, I mean, I have one of them left, but I worked on a lot of guitars. As in when I was an apprentice, I worked on guitars for several musicians in Canada. You may not know them, or people may not know them, but it was fun. I mean, um, I worked on a really expensive guitar that belonged to Leona Boyd. I don't know if you know Leona
I
Boyd.
don't.
She's a very famous classical Canadian guitar player. I worked on a couple of guitars that Bruce Cockburn had. Um,
Wow.
he had these two Flying V's, and I worked on them with a friend of mine. Uh, and there were other musicians, but you may not know. but it was fun. It was a fun time.
Do you still make instruments or guitars in your spare time? You mentioned you made that one behind you during COVID.
Yeah, so COVID, my daughter's a musician. She's a singer and a singer-songwriter. She also plays the bass. So I made her a bass a little while ago, uh, 3 years ago, I guess. And then I made her a ukulele,
Mhm.
uh, which is a sort of my first acoustic instruments that I'd made in close to 40 years.
Wow.
And I've made myself this Les Paul behind me. And I'm going to make another classical, um, sort of electric classical guitar. And I've had this idea ruminating in my head for a long time of it's going to be pretty unique and it'll be fun. And I have one funny story to tell you with regard to guitars because I'm very good friends with Arthur Christopoulos. I don't know if you're ever having Arthur as a.
He's busy. I've tried.
He's
I've tried. So
very, very busy.
Arthur, if you're listening, or Patrick, if you're listening, please relay the message to
Yeah.
Arthur. But yes, he agreed to come on the podcast, but he was busy because it was COVID, and it was this whole COVID situation, and he became chair or dean, I think, of the faculty.
Yes.
And
He is the
but I'm
dean of the
yeah,
pharmacy school in Monash.
yeah.
Well, not many people know this, but Arthur is an incredible guitar player. He's an incredible guitar player. He had to make the decision whether to be a musician or a pharmacist, and we're all glad that he chose to
Yes.
do what he did. But he has, I don't know, 25 guitars,
Wow.
and they're beautiful, and they're not just regular guitars; they're beautiful guitars. And he said it could have been one of those evenings, at a meeting sometime when everyone's relaxing and other things that he wants me to make him a guitar,
Okay.
an
Okay.
acoustic guitar. I said a steel string, and one day I will do that for him. He's gonna
Maybe
pay
the
me.
maybe the Great Lakes GPCR retreat, this meeting this year, is the place to deliver the guitar because he's going to be
Mm,
giving a talk, unless you
yeah,
have
no, it'll take a
take
while.
longer.
Yeah, I mean, if I were just making guitars,
Yes,
I
of course.
wouldn't, but I do have another.
Profession about which let's get back to it.
Yeah.
Thank you for sharing that. I think it's amazing. I think you're our first guest who's actually a musician and also a guitar maker. So it's
Yeah, I mean, I wouldn't call myself a musician. I always define myself as a hack. I'm an artist,
okay.
but I am an instrument maker. I do make instruments. Or I
I love it. I love it. Let's talk about instruments that you can only use in the lab to visualize structure, function, and relationships of GPCRs and G proteins. When did you first hear about GPCRs?
Oh, that was definitely in undergrad as certainly obviously in more depth as a graduate student,
Yeah.
but in undergrad, I mean, I was in pharmacology, so we had learned about drug action and
Mhm.
hormone action and how binding to the cell surface receptor would initiate and propagate these long signal transduction pathways, which have
Mhm.
some effect on physiology. So certainly then we were introduced to it. I never imagined, of course, at the time where I would be and in what kind of resolution we would start looking at those drug
Yeah.
effects. But that's when I first learned it about, certainly.
What was your first impression about GPCRs? And before you answer, I just want to mention that I first heard about GPCRs in Michel Bouvier's class as an undergrad at the
Yeah.
University of Montreal. And Alice Lapour was also I think he was doing his PhD in Michel's lab, and he took over some parts of the class. And I close my eyes, I can see myself, and I can see the projector. It wasn't even a projector; it was the old
Slides.
school slide exactly. And I'm like, wow, this is so cool. And we had the opportunity to work for a summer in any research lab. And my thought was, I want to work in this lab because they work on these really cool proteins.
Yeah.
What was when was that moment for you where you said to yourself, uh-huh, this is something I might want to get into?
When I throughout my undergrad, it was in pharmacology, but I got interested in membrane proteins because I thought that a lot of the biochemistry that had been done on an enzymology, and a lot of it had been done on, I don't want to say easier targets, but soluble enzymes
Yeah.
and soluble drug targets. And I just felt that the membrane was just introduced a whole new facet of effects. And when I started graduate school, actually, one of the first things I did when I started graduate school, believe it or not, was to purify a dopamine transporter. And so, in fact, I was able to purify dopamine transporter, and we were trying to get N-terminal amino acid sequencing data
Yeah.
from it. And this, at the time, it was only the GABA transporter that had been reported by Randy Blakely and Sunahara. And they didn't know there was a family of monoamine transporters. Although in retrospect, you can say, well, should have known that a lot of them is sodium dependent. It's probably the same, but it was really a different time. And so my starting off in Phil's lab, and I was working with Chaim Niznik. I don't know if you had a chance to know Chaim at all.
No, not at all.
Chaim was an amazing. He had a really large impact in my uh, initiation into GPCRs. So he was starting his career as an assistant professor, and he thought that um, he would try to open up the area of dopamine transporter biology, because he understood, yes, it's the target of cocaine
Yeah.
and other stimulants. And uh, it was tough, though. It was a different time, and we had faced the same challenges that Bob and Brian and Mark had faced when they were trying to purify beta receptor, except our transporter was expressed at significantly lower levels, and we didn't have the tools that they did for beta receptors.
Yeah.
So we ended up getting scooped um, in a big way. And there's nothing we could do about it. And the time in Phil's lab, Chaim Niznik was collaborating with Brian O'Dowd. And Brian came up with this really clever strategy, because he, obviously being a Caroline Lefkowitz trainee, and they had sequences of many of the GPCRs at the time, to come up with a strategy using degenerate PCR approaches to try to pull out other receptors. And so Brian designed some really interesting degenerate oligos to TM3 and TM6, which is, they're highly conserved
Yeah.
in many of the GPCRs that were known at the time. And then that's when I was following that, and I was thinking, oh, that's really cool. And I was following it while I was doing the dopamine transporter work, and I was thinking, oh, it'd be great if we could do that with the transporter. But yeah, but no, because we only had one version. We
Yeah.
didn't know there were family members, etc. So when we got scooped, we I thought um, I thought there was a real possibility that we can try to use the pharmacology that we had at our access in the Seaman lab to try to define the gene products that we could potentially get. And there was a very interesting PCR product between TM3 and TM6.
Mhm.
And because there were, Brian O'Dowd had identified a lot of different fragments. And there was one particular fragment, and the clone was called G36. I don't think anyone except Brian O'Dowd would remember that. And what was interesting about G36 is when we did a northern, we found that the message was in a lot of what would be dopamine containing neurons. And then, so I took G36 and ended up pulling out full length D1. And of course, expressing it and then characterizing it pharmacologically. And we knew it was D1 at the time. So my experience into GPCR was really came from membrane proteins in general. And I was just fascinated by them. And then I sort of, as I said, it was serendipitous, and started working on dopamine receptors. And then that's as the cloning era started to come out, we started to look at all the different sequences of the different GPCRs that were identified. And I had identified many GPCRs, because one of the first things I did was a low stringency homology probe strategy with the D1 clone. And I had pulled out, I don't know, about 90 different clones. And then as I started sequencing them and subcloning them, and this is, pre-PCR,
I was just
pre
gonna mention,
five
yeah.
$5 oligo PCR. Oligos are so expensive then, and TAC was so expensive, and it was really prohibitive.
Yeah.
And it also took a long time. But so you had to subclone everything. And in order to sequence things, I mean, we could only sequence
Portion of
400
it.
bases. So
Yeah.
we had to cut everything up into little pieces and then sequence those. And we had to sequence them ourselves. Um, yeah, and then it was sort of piling up and seeing all the primary sequences and how the primary sequences were able to uh, explain some of the pharmacology and some of the
Mhm.
specificity, some of them. And there was this classic paper by um, uh, it was a long collaboration that the Lefkowitz lab had with Merck.
Mhm.
And in particular, this work was even the first clone. It was a collaboration with Merck.
Yeah.
But there was this beautiful classic paper by Kathy Strader, who I think a lot of GPCR biologists should cite the work in lectures as a learning step, because Kathy Strader had, mind you, she was with a drug company, and so she had lots of drugs at her beta receptor ligands. They were able to basically figure out where epinephrine bound in beta 2, and this is
Wow.
pre-structure. I
Yeah.
mean, it was amazing. And to coupled with site-directed mutagenesis, and they had tried to generate three-dimensional structures based on bacterial rhodopsin, which are, of course, are all wrong. But the it was an amazing series of papers that the Strader Lab had published. And so what that excited me about was that, well, you can look at the sequences and you can explain a lot, but that's only the hormone binding site. And we have drugs which are sort of subtype selective, and they have this weird pharmacology. Three-dimensional structure must be incredibly important.
Yeah.
And at the time, this is in the 90s. Uh, there was no, there weren't any structures.
No.
Biology was really, really, really hard. And of course, moving to the Gilman lab and working on G proteins and adenylyl
Mhm.
cyclase, and that's when structural biology in general in the field became a lot more accessible. More people were doing it. Instrumentation was getting better. Um, um, Understanding of how to crystallize, how to isolate things, how to analyze the data, how to express proteins and how to express them recombinantly, and that had changed a lot. But and I think, after I um, left the Gilman lab, I wanted to go back to the membrane protein component, and um, started having fun with looking at beta receptor activation and beta receptor membrane dependence, and um, so that's how I got into it.
That's amazing. You mentioned, dopamine receptors and beta receptors. Do you have a favorite GPCR? It doesn't have to be a GPCR. Everyone who I invite, and sometimes they say, well, it's the Dr. GPCR podcast, and I work on beta arrestin or I work on G proteins. Andrew Tobin, for example, had a favorite phosphorylation site. So it does
Yes.
not have to be a GPCR, although the
Yeah.
question says,
But
what's your flavor?
his favorite phosphorylation site's on a muscarinic receptor, though.
Of course. Of course.
um, I beta two is my favorite because we know a lot about it. We have a lot of drugs,
Yeah.
but at the same time, we don't know a lot about it.
Yeah.
And so we have the tools to analyze um, more not far fetched, but deeper analysis of how it works. And I think that's been really fun. And we published a paper a couple of years ago on this is a collaboration with Brian's lab, to figure out how norepinephrine can distinguish between beta 1 and beta 2. I don't know if you beta 2 has a very poor affinity for norepinephrine. And if you look at the crystal structure, you couldn't predict why it would. It was just, it didn't make sense. And of course, if you look at the primary sequence, just as I said, when I was in graduate school, looking at primary sequences was completely uninformative,
Yeah.
other than the fact you knew what were important for the actual orthosteric binding site. But it's because we had a lot of tools at our hand, and we had a very much a better understanding of how the beta 2 receptor worked, that we could apply it to the beta 1 receptor. And then it really helped us understand and how to interrogate and how to interpret the different, in this case, turned out to be kinetic differences between binding to beta 1 and beta 2. And that turns out the kinetics, classic kinetics, receptor binding off rates and on rates, and couple that with mutagenesis and couple that with certain different properties of the orthosteric ligands itself, helped us to understand why norepinephrine works better on beta 1 than beta 2. And again, it's because of what we know about beta 2. And so that would be my favorite receptor because of what you can do with it, I guess, the kind of deeper questions we can ask. My favorite receptor, which I have not worked on since I was in graduate school, is obviously dopamine D1 and D5 receptor, because we were the first to identify them,
Mhm.
or at least D1 together with Olivier Civelli and with Mark, and then D5 we were far ahead of the other guys.
Mhm.
But I've always been interested in going back to study it. Only because I could learn, because the D1 and beta 2 are actually pretty similar in many ways. So I can
Mhm.
sort of pull from what we know from beta 2 and try to extrapolate it and put it on it as a template for how D1 would work. But
When do you think you'll ever be working on the D1 or maybe D5 receptor in the near future, now that you have a well-established lab and it's been over 10 years at UCSD?
I think I'm I've been more interested more recently in the distinct signaling pathways that are involved in GPCR biology, using the beta 2 as an example. And I think we've, not to say that we know everything, but we've learned a lot about how basically beta receptors work,
Yeah.
the basic function. How does it, what are the conformational changes when agonists bind? What happens to it when G protein binds? What happens when arrestin binds? We have pretty good, although not complete, but pretty good models for how they work. And I think what we don't have a lot of detailed models are sub downstream signaling.
Yeah.
Cyclase, yes, we have pretty good, but the rest of it, we're pretty lost.
Yeah, I feel like
Um,
with all of the all GPCRs, we kind of figured out which receptor does what. We know some G protein, some beta arrestin signaling, but it's way more complex
yeah.
than these
And so
three
what I
partners.
would like to do is continue on beta 2 on looking at the downstream signaling, and then be able to look at D1 and in its downstream signaling, because it is much more complex. Dopamine receptors have a very interesting downstream signaling pathway. And Marc Caron, I mean, he spent years and years working on D2 signaling, and it's really, really complex. But I think we've generated interesting reagents that can interrogate some of these pathways, and not just reagents, but also techniques. I have this really, really fun collaboration with someone here at UCSD, Jen Zong. She's a reporter guru in signaling. She has amazing collection, developed an amazing collection of reporter assays for um, um, second messengers as well as phosphorylation. Probably her largest collection is on phosphorylation reporters, kinase reporters. And we've had a lot of fun working on beta 2 regulation of ERK, for example. And we had a paper just published last month, and it introduced a lot of new ways to look at receptor activation, and it unraveled a really controversial area of the contributions of arrestin towards ERK activation.
Mhm.
And we're obviously going to continue to expand on these and look at other cell lines and look at other sort of models. But what it did is it placed a clear dependence on arrestin, that you required arrestin for
Yeah.
endocytosis, but that the ERK activation occurs on endosomes. And that was sort of a surprising, that it occurred on endosomes, but not at the plasma membrane.
Just I
And so
think
that,
it's
yeah, that
still
turned out to sort of change how we
yeah.
we can look at things.
And because when you think classically about the activation of a GPCR, you do have ligand binding and then G protein activation, then phosphorylation by GRKs, and then beta arrestin recruitment, and then endocytosis. And
Yeah.
then sometimes we stop there, but we know that's not the end of the story. And I think being able to differentiate the compartments in which some of these receptors activation leads to. I'm thinking here about the prolonged cyclic AMP activation of the PTH receptors,
Yeah, same thing.
which I think is a really
Endosomal,
fascinating.
yeah,
Yeah.
it's the same thing. But now I think the field is starting to develop all these tools to be able to look at that. And I think I find that fascinating, because it's beyond the realm of conformational changes per se, and you can really start to see. Yeah, I mean the second wave of or prolonged activation of PKA and cyclic AMP production in endosomes, I think, is fascinating. I mean, that's how we started actually in ERK, because I collaborated with Mark Monzastro on the
Yeah.
original paper by Roshana Karanashad, because we had some really useful reagents to try to dissect that pathway out. our nanobodies to the beta receptor and GS alpha.
Yeah.
And so we could, Rashanik was able to look and see that the beta receptor is actually in the active state in endosomes, and that the G protein GS gets activated on endosomes, and that that component, that activated G protein on endosomes, is the thing that activates adenylyl cyclase, and it's the endosomal compartment which is responsible for regulation of Krebs and transcription.
That's fascinating. I wonder if and this is a completely naive question, I wonder if we've ever been able to tie endosomal signaling to a disease state. And if anyone has ever been able to say there was a change in signaling and now it's signaling from an intracellular compartment as a consequence of a disease state.
Yeah, I mean there are hints of it, certainly. Uh, endosomal signaling is not new at all. I mean, it's been studied for years on EGF receptor activation,
Yeah.
and also just regular, all the RABs have been characterized for years for membrane
Yeah.
replacement, just membrane homeostasis. And there are many point mutations in a lot of the RABs, et cetera, and interacting proteins that have been involved in disease, implicated in disease. Now, the direct connection to receptors and GPCR signaling, I think we're only now do we have the tools to look at that. And so I'm pretty sure people are going to be interrogating that.
Probably, probably. I always ask this question and 19, well, actually 100% of the time the answer is yes, but I always ask my guests to expand on it. The question is, do you think GPCRs are still good drug targets?
Sorry?
Do you think GPCRs are still good drug targets?
I think they're excellent drug targets.
I think so
Not
too.
just good. I think they're excellent drug targets. I think that we um, still need to learn more about it, but I think one of the challenges the field has faced, and certainly the pharmaceutical industry has faced, is making subtype selective drugs.
Yeah.
And now that we know a lot about structures, and we have to think that in order to make subtype selective drugs, we had to go outside of the hormone binding site. And indeed, allosteric modulators have really been able to do that and accomplish that task. And now allosteric modulators as drugs are new.
Yeah.
The concept in general is sort of new, in a grant, on an average. Of
It is
course,
because it's difficult
yeah, it's difficult, and you have to have the right kind of cooperativity,
yeah.
to be able to make the drug, but also make a good drug that has all the physical chemical properties that you would expect to be a good drug, and bioavailability and distribution and et cetera, et
Yeah.
cetera, et cetera.
Yeah. And then
I
again,
think,
depends where the receptor is also expressed because sometimes you want to modulate the receptor in a tissue versus another one. And then how do you direct the drug to the specific tissue?
yeah, I mean, I think there's a reason, even though we have a lot of sort of crappy non-selective drugs,
Yeah.
it's still 33 to 35% of all
Yeah.
drug.
Yeah.
So I think our ability to make safer, more efficacious drugs take into consideration other parts of the receptor, are are the future, and they will certainly help.
I think so too. I think there's a key need for more tools to better dissect and to establish the relationship between GPCR expression in specific tissue, its signaling, and its relationship to the disease in order to better modulate and not necessarily turn on or off the GPCR.
I mean, GPCRs have a lot of advantages. compared to other targets.
Yeah.
It's at the plasma membrane, usually at the plasma membrane. So access to the cell and the drug target is inherently easier.
Yeah.
Um, the target, the at least the orthosteric site is usually uh, uh, is such that you can have drugs that are will work that can still bind. In other words, that it has the right partition coefficient, it has the right size, and it a lot of the Lipinski rules
Yeah.
can be followed for ligands which target the orthosteric site. Now, the allosteric site is a bit more challenging, but I think
Yes.
GPCRs do have an advantage that they have a pretty nicely defined binding site. So you can create high affinity ligands, at least to the orthosteric site.
Yeah.
And they get into the drug target without having to, permeate to the cell to get into the nucleus or whatever.
Agreed. Agreed. All right.
I
Last segment. What are or are the top three aha moments that you had as a scientist that shaped your trajectory?
I have been lucky in my career. If I could narrow it down to three,
Can be five as well.
well, the first one was um, when I'd cloned D1 and expressed it in the very first binding assay I did.
Mhm.
This is one of these classic experiments where it's like it was 3 in the morning. I was there, and Heibniz came down because he knew I was testing it at 3 in the morning, and we were waiting for counts to come off the
Yep.
counter, and
Yeah.
it's so slow.
Yes, and then the printer sometimes jams up and
Yeah,
then runs out
yeah,
of paper.
yeah. No, and when I saw that first binding assay and the total and the non-specific, and then I knew I had it, and it that was an amazing experience, and we were high-fiving and jumping all over the place,
Yeah.
and that was really fun. Um, the second, uh, I wouldn't say it's a eureka moment, but it was a moment when I went down. This is a Southwestern. I was a postdoc, and we were going after John Tesmer and I were collaborating, going after the crystal structure of adenylyl cyclase bound to GS, and we had a number of different crystals, and we were getting various diffraction. We had one really good data set, and John was just sort of figuring out. And I was down at the graphics terminal with him, and it was just figuring out, and he solved it. And just to see the structure of adenylyl cyclase, the catalytic core adenylyl cyclase, where GS bound, what part of switch 2 was bound, where it bound on cyclase, and it was amazing. It was really amazing because we worked really, really, really hard to get that structure, and we knew it was going to have more broad implications because it could affect other adenylyl cyclases as well as guanylyl cyclase because the enzyme's essentially the same. So the catalytic mechanism would be the same. But it was really amazing to see that structure. Um, if I were to say there were a third uh, discovery, this is in collaboration with Brian Kubilka. So we had developed this uh, reconstitution method to study GPCRs in lipid. And because it was so efficient, we were able to basically reestablish all the correct properties of isolated receptors, um, and it would display the correct pharmacology. It would display the same pharmacology that would see in membranes. So typically, detergents perturb
Yeah.
ligand affinity and ligand binding, and it was a problem because you had to think back about crystal structures, and crystal structures are all, at the time, were all done in detergent micelles. And we knew the importance of lipid. And this set of experiments actually started, the idea to do these experiments started when I first really started to talk to Brian. And this was at a Gordon conference in 2005. And I dragged Brian over to my graduate student's poster, where I was showing him this method that we developed to reconstitute and look at G protein coupling. And there were two things which he couldn't believe: is how well behaved the receptor was, but also he couldn't believe how efficient our G protein coupling was. And so, we were talking throughout the meeting, and then, Friday was the end of the day. We both went home, and then Saturday we emailed back and forth and back and forth and back and forth. And Sunday we sort of had meetings with our labs, independent labs, not
Yeah.
in-person meetings, but
Yeah, of course.
because it was Sunday. Actually, I had my graduate student was in, so I went and talked to my grad student, and then called Ryan back, and we sat out a strategy. Monday, he purified some beta 2 receptor and labeled it with a fluorophore. He sent it to us Tuesday. We got it Wednesday and reconstituted it and did all the basic pharmacology, and it was all exactly like how we expected it. And this is fluorophore labeled beta 2 receptor. And the reason why it was so interesting was that this fluorophore that Brian had, the site labeling beta 2 is labeling a specific cysteine in beta 2 on TM6, was really, this fluorophore was sensitive to environment. So when TM6 moved,
Oops.
you can get a change in fluorescence. Now, the problem that Brian had in the lab at the time was he was doing all this in detergent micelles, and the kinetics of activation was really poor, because basically, receptors hate detergent. And so the halftime of activation of our reconstituted receptor went down to like diffusion limited activation. So we saw a halftime, and we didn't have a mixer, a rapid mixer at the time. And we saw activation in like, it was basically 6-second activation,
Wow.
which we'd never seen before, because it was in the minute time scale in detergent micelles. And so that was a pretty much of a eureka moment, because that thought, oh, finally we have a preparation where we not only have a preparation, but we have an assay to measure real-time conformational changes in the receptor as it gets activated.
Yeah.
And this was coupled to another study that Brian and I had worked on. It's not TMR labeling. It was a bimane labeling, which is a different fluorophore. It's much smaller. Bimane is around the size of a tryptophan, so it's not as sterically impinging on receptor activity. TMR is huge. It's like fluorescein. They're just big, bulky, aromatic compounds. Bimane is about the size of a tryptophan residue. And so when we first translated all of that to bimane labeled receptors, we could see the conformational change. We can see obviously in three-dimensional structure, but we can look at the data and see what a full agonist does, what a partial agonist does, what a neutral antagonist does, and importantly at the time, what an inverse agonist does. And we could see it. And that for us was huge, because now all subsequent experiments, if we had to screen reagents, if we had to screen mutants, if we had to screen anything, we could always use that assay to measure it. So nanobodies, all of our nanobodies, we characterized using bimane labeled receptors. And the nanobodies are incredibly important for structural approaches later.
Of course.
So that was probably for a third really important sort of eureka moment, if you want to call it that. And of course, the fourth was seeing the structure of the receptor G protein complex.
Of course.
That was again, that's something we had worked on. We were working on for years and years and years. And as we started getting better crystals, um, so this is when I was at the University of Michigan, and we used to collect our data in Chicago and at APS.
Yeah.
And we needed time. We needed detector time, and it was really hard to get detector time. So what we were able to do is get detector time when no one else wanted it. And it would open up they'd say, oh, well, so someone's not, you got 24 hours, you got to get to Chicago and you can have this 24 hours. And so we would get a lot of those. We are fortunate, extremely fortunate to be able to get that time. But also because I lived in Michigan, we could just pick up and
Yeah.
drive there
Yeah.
and get in. And so we had every week I was in Chicago collecting data. And in one of these weeks, I was there and Andy Cruz
Mhm.
was there and we are collecting data, and Andy was collecting data on muscarinic receptors, which ended up being critical to his M1 and M2 structures that he published a couple of years after. But also, we were collecting data on the complex, and uh, we had these amazing diffracting crystals, and we collected some really nice data. So we couldn't solve the structure. And one of the reasons why we couldn't solve the structure, it's because we were trying to use heterotrimer as a so we were trying to solve it by molecular replacement and trying to use
Yeah.
the heterotrimer as a search model. The beta receptor was different enough from the activated beta receptor was different enough from inactive beta receptor that we couldn't use that as a search model. So when you used heterotrimer, it didn't work, and it just didn't make sense to us. Like, why can't we use heterotrimer? And then so we said, okay, well, let's break it down. Maybe we can use alpha or beta gamma.
Yeah.
We used alpha alone, didn't work. Used beta gamma, popped out. And
Wow.
we saw the whole structure, and it was amazing to see. It was amazing. The drag about it was just, Brian and I had been working on this thing for years, and he wasn't there. He
Wow.
wasn't there. We couldn't share the moment together. He was in China, and so but immediately we
Of
Skyped.
course.
We're talking about it, and it was an amazing. That was amazing. Uh,
Good times. Good times. And you were lucky that because you could just quote unquote drive there at almost
yeah,
any moment,
yeah,
at a
yeah.
moment's
I
notice.
mean, my family hated
Of course.
it
It's
because I would leave at Friday at you know 6 o'clock, and then they wouldn't see me until Sunday.
wow.
So I'd leave Friday p.m. We get there um, just before the gate closes because there's a security gate, and then we would get time at 12 o'clock. That's when the clock would tick, and then we would collect data for 24 hours straight.
Wow.
Uh, and we tag team who would actually be there
Yeah.
collecting, mounting the crystal, et cetera, et cetera, et cetera. So it was an amazing time actually, and I many times look fondly upon those moments, and other just because so diffraction was a big point. So we had diffracting crystals for a long time, but they were like 12 angstroms, and we couldn't do anything with the 12 angstrom
Of
structure. Well,
course.
we could. I mean, it would be informative, it'd be novel, but we really wanted an atomic resolution. And the time when we broke that, the structure really quickly fell out. And but so many weeks it was stuck at 12 angstroms. We have these new crystals under new conditions with new
Yeah.
nanobodies or new something, and then we'd set it up. We drive all the way to Chicago and do everything, and it's 12 angstroms again, and
Wow.
repeated. Week later, 12 angstroms. A week later, 14 angstroms, and then it sort of just popped out. We
Yeah.
got this. In fact, our best data set, even though we couldn't, we never published the data to that resolution because we didn't have a complete data set. But we had our best crystals were probably about 2.6 angstroms resolution limit. And it
Wow.
was just one of those unfortunate times when we had this beautiful crystal that well, it's sort of hard to describe, but in the hutch where the um, detector is,
Mhm.
there's a video monitor that looks at your crystal
Yeah.
just to make sure that it's in the right grid and in the right view. And when we were strategizing on how to collect the data, it was Andy and myself and Tang, who was a postdoc in Brian's lab, and we were sort of strategizing on how we were going to collect this data on this big crystal we had isolated. We collected some data from it,
Yeah.
but we wanted to collect a full data set. We watched the thing in the monitor start to shake,
Oh God.
and then we saw these basically snow ice just like pummeling our crystal, and we watched it
Getting destroyed, basically.
destroyed the rest of the crystal. And this crystal was beautiful, and we were never able to get a crystal as nice. We got several crystals before, but not quite as nice. And if for not that problem, that I think our receptor G protein complex structure would have been, probably 2.8 angstroms
Wow.
instead of 3.4. And that makes a big difference when you talk about uh, side chain differences. And
Yeah.
but anyway, so that would be my fourth sort of eureka. It was really amazing to see it, and it was also amazing to talk to Brian about it because we were just so happy.
Of course. Of course. I remember
There's this
Fiona
there's this
Marshall telling. Sorry, please go ahead. This is
there's
a
this
delay.
pic, there's this picture that Brian shows all the time. I used to show it as well, and it's a picture that Brian took, um, and it was after we had collected our last high resolution data set. So we basically had the whole structure, and we knew what it was going to look like, and that was the structure that we were going to publish. But it was the last data set, and I drove them to the so my grad student was there, Soren Rasmussen was there, and Brian was there, and I was dropping them off at the airport.
I saw the picture. I've
Yeah,
seen it. I've seen
yeah,
it.
that was
It's a
it,
great
and
one.
and you could see how happy we were because it was just like that was it was an amazing time, a lot of fun.
I remember, and I think I've seen the picture in the context of one of your presentations, but I think Brian also shared that picture, and you can see all those smiles, the satisfied smiles when the last data set was acquired.
Yeah, I mean, for me, and certainly for Brian, that was like a 14-year project.
Wow.
I started the complex that we solved is something that I had started basically in the Gilman lab. So, I left Al's lab in 2000, but I had started it like 1998, and then continued working on it.
Wow. Well, I
So
think I think this is a great example of perseverance where you know that you shouldn't stop. There are difficulties, but then there was an end goal. So we were talking with another podcast guest about, is there a moment in your project where you tell yourself, okay, I need to move on because I have a limited amount of time, a limited amount of resources, And what do I do? When is that time to say I'm done with this project and I'm going on to doing another one? But sometimes if you just keep at it, you'll get there eventually.
Yeah, so I've played sports all my life, team sports, and there's this term that actually my old boss used to use it, Al Gilman, is you got to know when to punt, as in
Okay.
football terms. You really have to know because you could you could bang your head against the wall so many times and you just take a layer of skin off your forehead one at a time and just you keep doing that, you're not going to have a forehead left. And that you have to do. I mean, when I started into GPCRs, and then not that many people know this about my trajectory. Um, I was a I wanted to study GPCR oligomerization.
Mhm.
We had some data which suggested that dopamine receptors can form oligomers, and we were uh, intrigued by some of the pharmacology that we had on D2, for example. And I really felt that dopamine receptor D2 oligomerization was going to be a huge thing. And I went to Al Gilman, and I was proposing a project to him on dopamine receptor oligomerization. And I gave him all my data and showed him why I thought this and the various models. And well, he said, "Oh, that's very interesting. That's really, really interesting." And I said, "Yeah, and I think GPCR oligomerization is going to be the biggest part of GPCR biology." And he turned to me and he said, "That's so interesting. And you're probably right. I think it's really going to be a big future of GPCR biology." And then he said, "But this is what I want you to work on." And I ribbed him about that. And I worked on adenylyl cyclase and
Yeah.
things that were now, but he explained years later why he said that. Um, he said that oligomerization is really hard to study.
Yeah.
And it's really hard to study in a cellular, particularly in a cellular context, and for in much less in an organism. And you have to be able to do things that are tractable. It's just especially when you're
Yeah.
starting as assistant professor.
Yeah.
And in my very first grant proposal, I had actually proposed to look at receptor oligomerization
Mhm.
um, as one of my aims. And I I sort of realized how difficult it was to study receptor oligomerization. I had very naive views of how to look at it in vitro. Well, they weren't naive in vitro. It just the naivety came from, if we can see things in vitro, then it must happen in a cell or in
Yeah.
a. And then it sort of dawned on me, well, wait a minute, we're creating this artificial environment in vitro.
Yeah.
Um,
Yeah.
maybe it's not the right thing to study. And so for the rest of my career, I decided that I wasn't going to study oligomerization. I was going to study the isolated monomer by itself to see how it behaves. And so I consider that as a punt because I just realized the complexity of how to look at receptor oligomers. And I would love to be able to go back to it and try to figure it out. A lot of people, from the direction of my research, a lot of people in the field have thought I was anti oligomerization. And I'm not an anti-oligomerization person. I just don't study it because I think it's really, really difficult to study, and we don't look at it. We don't use a lot of the approaches, at least not routinely, to look at
Yeah.
them.
Yeah.
And
I
so
think I'm
I
sorry.
should probably dedicate my efforts to looking at the simplest model. So
It's interesting because you were mentioning oligomerization. I think, especially at the time where you were proposing to work on a difficult and very risky project, we didn't have enough tools
Yeah.
to study oligomer. And I'm not even sure if we have the right tools now to look at receptor oligomers in native tissues.
Yeah, I think imaging approaches, I think single molecule spectroscopy is the way to look at it. And I think probes are better, and people have certainly been able to do it in cells. And I think that it's not too far to be able to look in tissues, especially with the two photon microscopy and et cetera, et cetera. So I think it's closer, but it's still, for me and what I do and how I can contribute, I think it's better that I spend my time trying to figure out how a simpler model works, and then before I can build anything bigger. And so,
amazing. Thank you so much, Roger, for your time. I really appreciate it. I know we're at time. Please don't go anywhere. I'm just going to stop recording. But it was a great pleasure having you on.
Well, thank you. It's been a pleasure. Really a pleasure.
Thank you.
Thank you for joining us and listening to this Dr. GPCR podcast episode. We would like to thank our guest, as well as take a moment to thank our team members, Attila, Ines, Monse, Ivana, Andrina, Balint, and Julia. A huge thank you to our ecosystem partners for their support: Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, Montana Molecular, and Orion Biotechnology. You can connect with our partners directly in the ecosystem, so don't forget to join the Dr. GPCR ecosystem today. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcasts. You can also leave us a testimonial by following the link to the testimonial forms in the ecosystem by using the footer. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions and suggestions at hello@drgpcr.com. And until next time, stay safe.